Hi guys, welcome to the second video in the Understanding Cancer series. So in the last video we left off talking about E2F and cyclin E. So just as a refresher, some of the important targets of E2F that we're going to discuss in this video were cyclin E, CDK2, cyclin A, and also DNA repair proteins, MCM and ORC. So as we said in the last video, cyclin E is involved in a positive feedback loop because it phosphorylates when bound to CDK2 and activated. It phosphorylates PRB, thereby releasing E2F and upregulating its own transcription because it is a target of E2F. And so what also happens is that this cyclin E and CDK2 complex, when the concentration of this increases in the cell, it begins to phosphorylate itself, other cyclin E CDK2 complexes. And once this complex has been phosphorylated, the cyclin E detaches from the CDK2 and it is targeted for degradation through ubiquitination. And so that's out of the picture. And this now allows cyclin A to bind to CDK2 because the CDK2 is now free. So cyclin A competes with cyclin E for CDK2. So this cyclin A CDK2 complex is important because what it will do is it will, as its concentration increases, it will begin to phosphorylate E2F. And once E2F is phosphorylated, it causes a conformational change in E2F, rendering it unable to serve as a transcription factor. So this essentially halts the production of cyclin E, cyclin A, CDK2, and the other proteins such as the repair proteins. So this is a way of allowing the cell cycle to continue. The cyclin A CDK2 complex is also important because it is what is responsible for initiating the pre-RC or the pre-replication complex. So the pre-replication complex is composed of the ORC, which, as we mentioned earlier, is a target of E2F, and this ORC is a protein that is responsible, well, it stands for origin replication complex. And it binds to the origin, and it is composed of 6 protein subunits, which kind of serve as a base for other proteins that will come and bind to it and form the whole pre-RC complex. So one of the proteins that will bind to the ORC is called CDC6. CDC6 stands for cell division cycle 6, and it is another protein that will make up the pre-replication complex. And the binding of CDC6 is important because CDC6 recruits CDT1 and allows it to bind to the ORC complex. And so we'll demonstrate that down here as this complex binds, thereby completing the pre-replication complex. So here we see CDT1 is important to the replication complex because it now allows MCM proteins to bind. And MCM proteins are also targets of E2F, as we mentioned earlier, and they form helicase essentially. So the MCM proteins 2 through 7 actually form a hexamer on both sides of the ORC complex. I'm only putting it on one side, but they're on both sides, and they begin to unzip the DNA from both ends, thereby forming the replication fork that you normally see at origins, the double replication fork. So now the cyclin A CDK2 complex becomes important now that the helicase has attached to the DNA. And so what actually happens here is that this cyclin E CDK2 complex comes here and it phosphorylates the CDC6 and the CDT1. So both of these are phosphorylated, and this causes them to detach from the ORC, not quite like that, detach, and either be taken out of the nucleus or be targeted for degradation through ubiquitination. And what this essentially ensures is that replication only happens once, because CDC6 and CDT1 are needed for replication to happen. So if you get rid of them, replication can only happen once. So this is kind of a safety measure. And finally, the last step is for the cyclin A CDK2 to phosphorylate the helicase itself, the MCM hexamers. And once they are phosphorylated, this is like the go signal, and it tells the helicase to begin unzipping the DNA, and S phase begins.